if you learn if you learn about general concept, general characters of algal member, you can understand any class of algae. Chlorophyceae class type studies, xanthophyceae class type studies, Phaeophyceae, Rhodophyceae class of type studies. Before enter the type studies, you should aware about the general concept. So that's why in this context we completed from the first class algae general character, right? So based on the general character, you should read each class chlorophyceae type studies. A chlorophyceae, I think given Volvox, one of the type study, Chara, Hydogonium 3, Xanthophyceae, Wachiria, maybe. I think Wachiria, only one class, only one type study, Wachiria. Phaeophyceae, Ectocarpus, Rhodophyceae, Polycyponia. So these type of type studies are given. Just read individually these type studies based on the general concept that already completed clearly in last class. So I request you all, one and all, please go through the general concept of the algae. While you read general type studies of algal member, just compare, just differentiate with general character. Please go through the general character. You can understand easily. If you have any doubt, any complexity, just ask me. I will explain. Okay. I don't want to waste your time and my time. Maybe I don't know how many classes are completed in offline classes. So it is very difficult to complete all the class, all the syllabus in online. So that's why just online classes, I will complete only those are important classes. That means the important means that is not only in examination, that important for your subject. For example, algae general character. If you really develop the concept of general character, you can easily understand complete algal type study. No doubt at all. So in this context, now the today class is bacteria. The bacteria in it. The very important bacteria important and virus also important. So now I'm going to complete about the bacteria class. So please remember those words what I told just now. The read yourself about the type studies, algal type studies. What are the remaining algal part? Just read. If you have any doubt, ask me. We will discuss the next class. You already
Okay, done,
read
sir.
it. You ask me now. Immediately I will clarify.
Okay,
Okay,
sir, and what
right.
about Unit 1 and Unit 2? So, we also need a summary on that again.
Okay, what is the unit one?
Let me check, sir, one second. Like, I'm outside
I
actually,
think
so
I think origin of life and biological timescale unit one.
yeah, yep, sir, correct.
The biological timescale anybody should memorized. That means there is
Okay,
no explanation because there are only era and periods. You should by heart era. You should by heart periods, and also you should by heart what period, what organisms are there, what organisms are existed. So there is no scope to explain. So anyway, you read it, and if you have any doubts, ask me. I will clarify. First unit
okay, sir.
also. What is second unit? I think virus. Second unit. Third unit bacteria.
Yes, sir.
Right. Now let me complete about the bacteria. While I explaining the bacteria, just compare with virus also. Okay. Right. Just wait. Let me share. Where it is. Share PPT. Just wait. Is it visible? PPT slide visible to you?
Yes, sir.
Right. So the very important question in bacteria unit, in bacteria unit, you should read each and every topic. The first most important one topic is bacteria ultrastructure. Bacteria ultrastructure means bacteria cell structure because it is unicell. Next important question is bacteria reproduction. Next important question is economic importance of bacteria. These three are very important. So bacteria structure, that is based on only the diagram. Reproduction also based on diagram. But economic importance you can read yourself. You can read yourself. First you read it, and if you have any doubt, ask me, I will explain. First, anyway, in bacteria, let's complete about the bacteria cell structure. Okay. Already told you while I explaining bacteria, I compare with virus also. So, what about virus cell structure? Is there any cell structure to virus? Hello? Any particular cell structure to virus? Hello, please respond. Any particular cell structure to bacteria? Please be silent. Hello. Please respond.
Yes, sir. DNA, RNA, like bacteria, na, sir.
Yes, I am asking bacteria first.
Okay, so DNA, sir, like you'll have a DNA, like membrane wall, like surface layers. So
What about cell wall?
So
Cell wall present or
flagella.
absent? Cell wall present or absent?
Cell wall is present, sir.
cell wall is present, sir, present in bacteria.
Right. But in virus, there is no clear cell structure. There is no cell components, no cell wall, no plasma membrane, no slime layer. They are all completely absent in virus. There is no proper shape also. In virus, just only the protein variants and also nucleic acid there. Only nucleic acid and covered away protein. That's it.
Yeah, yes, sir.
That
Either
nucleic
RNA or DNA.
rice, any one only, not one, but not two, but not both together.
Yes,
Either
sir.
DNA or RNA, any one present in a virus. But in DNA, the clear proper cell structure present with with distinct cell wall, with distinct plasma membrane, and both nucleic acid present, DNA present, and also RNA also present
Yes, sir. In
Sir, in
bacteria.
bacteria, sir.
in bacteria. So yeah, let's
Okay.
complete the bacteria cell first one by one. This is the bacteria cell. Is it visible to you? Hello?
Yes, sir.
Right.
Visible. The
So
ultrastructure of typical bacteria.
right, if you see this bacteria, the bacteria is the prokaryote cell. What is difference between prokaryote and eukaryote? Difference between prokaryote eukaryote?
So prokaryote is like so where you'll have all this nucleate, like I mean DNA, rhizomes, like cell structure, cell wall in prokaryotes. So
Okay. Eukaryote?
like only a single, so only DNA or RNA. Like in viruses, example virus.
Eukaryote example virus.
Yeah, I mean it will have only RNA or DNA. No, so
In
Sir, in
eukaryotes,
prokaryotic cell, the two nucleus is absent, sir, which is present only in eukaryote.
what is present in only eukaryote?
Nucleus, sir.
Nucleus present in eukaryotes, okay. But in prokaryote?
Absent.
uh, DNA, rhizomes, cell membrane, cell wall, everything. So like surface layer.
Okay, if you right, uh, where is nucleic acid? Look, where is the presence of nucleic acid in any organism?
In nucleus.
Yes, in nucleus. But nucleus present or absent in bacteria?
Nuclear
Present.
DNA is there, sir. DNA is there in bacteria, sir. Nucleoid.
Present.
Okay, but according to you, you said in prokaryote nucleus absent. Nucleus absent means there is no DNA, there is no RNA, right?
I didn't say,
Yes.
sir.
But of course, in earlier books must have given as a nucleus absent in prokaryote,
Yes.
but somewhat wrong that statement. Why? Because actually
Okay.
nucleus not completed absent in prokaryote. Nucleus, some of the components absent. The sum of the components means if you see this bacterial cell structure, you can observe the nucleic acid, nucleides. Here, this is the genetic material such as DNA, RNA, right? This structure. Are you follow my marker?
Yes, sir.
Hello?
Yes, sir.
Right. Is there any cell membrane around it? Is there any cell membrane around it?
Yes, sir. Like in bacteria, there's a cell membrane, sir.
No, that I am asking only for genetic material, not about the cell. I am not asking about the cell. Only this genetic material. Is this genetic material covered by any membrane? Only genetic material, not about the cell. Don't get confused here.
Okay.
Just wait.
No, sir. Like genetic material, there it has only like DNA, no, sir. So
Yes, only DNA. My question is there any protection layer to DNA? Any,
nothing,
are
sir.
you, nothing,
Inside.
nothing. Only the genetic naked DNA material present without protection, right?
Yes, sir.
That's what the prokaryotic cell. In prokaryotic cell, the clear difference is a nucleus. Nucleus means nuclear membrane absent only. Nuclear less also absent of the nucleus component, but genetic material present. Genetic material is a part of the nucleus, right?
Yes,
So
sir.
only genetic material present without protection layer, that is naked genetic material present, that is called as nucleide, which is the genetic material made up of DNA, right?
Yes.
And apart from that, only 70S ribosomes, 7CS ribosome there, and in cell structure, plasmid also present. Here the ribosome, pili, flagella, slime capsule, nucleide, already told you, cell membrane, and cell wall. These are all
Okay.
present in bacteria cell. So
Sir,
let's
we are not
discuss.
able to see PPT, sir. Like, it is only stuck on the ultra structure of typical bacteria cell. I'm not able to see other slide.
Just wait.
Okay, sir.
Yes, sir. The first page is showing.
What you observe now? Do you observe?
Yes,
Are
sir.
you
In the
observing
text
the
book, we observed, sir.
bacteria diagram visible or not?
Yes.
No, sir. No, sir.
Just wait.
So not in the PPT, we are looking in the textbook, sir.
Yep. Now it's visible, sir.
Which one visible?
The bacterial cell.
Yeah.
The third slide.
Now visible?
Yes.
Yes, sir.
Bacteria cell, the bacteria cell and with the diagram.
Yes, sir.
Clear?
Yes.
Okay. So in bacterial cell, we can clearly find out the first slime capsule, that is the outermost layer, and next cell
Yes.
wall.
Yes,
Next
sir.
to this slime layer, cell wall present. After
Yes.
cell wall, please remember the order. Please remember the order.
Yes,
Plasma
sir.
membrane, plasma membrane. After the plasma membrane, the all cell organelles, nucleus present. This is the nucleus such as naked genetic material, such as called as nucleoid, and also plasmids also present. Pili, flagella, they are the cell components. Let's discuss one by one. Okay.
Yes, sir.
Okay, sir.
So this, this diagram is the microscopic view of the bacteria. It's not important. So are you able to see bacterial shape? Bacteria come in wide variety of shapes. Which slide visible to you?
So the second slide.
Second slide means
This is a diagram of a typical bacterial cell. That one.
bacteria wide range, wide variety shapes, spherical, rod shape, DNA like elongated. These are all visible or not? The only words.
No, sir, not really.
Now visible.
Yes, sir. The cell wall.
The cell wall. Okay, just wait. Cell wall. Slide visible or not?
Yes, sir.
Right.
Visible.
Actually, before cell wall, actually here, uh, the shape of the cell wall, some not such a important topics. The shape of the cell wall, globe, spherical shape, rod shape, Wimbrier shape. So the such a different type of bacteria also there. That is not important one also. But here
Okay.
the important one only cell wall structure. One by one, let's complete one by one. First, actually before cell wall, first we have to complete about the slime layer, because this is the
Okay.
outermost layer of the bacterial cell.
Okay.
But this slime layer not present in all type of bacteria. Only few bacteria have the slime layer. Or
Okay,
this
sir.
is called as capsule. Slime layer or called as capsule. Okay, right? So, but first we discuss about the cell wall. The cell envelope is composed of the plasma membrane and the cell wall. So actually, cell envelope, not only plasma membrane and cell wall, apart from the cell wall and plasma membrane, another layer, slime layer also present in some organisms, some of the bacteria.
Okay.
So the bacteria cell wall provide the structural integrity to the cell. So it gives the proper shape to the bacteria. Whatever its shape, maybe it's spherical, maybe globose, maybe comma shape, maybe S
Sir, the
shape.
capsule.
What?
Capsule gives the shape, sir.
Yes. Please, can you repeat again?
Capsule gives the shape, sir.
What? Which shape?
The cell.
I'm not getting understand. Are you talking about shapes of bacteria?
Yes, sir. Yes, sir.
So already told you spherical shape, rod shape,
The voice
vibrio,
is not
comma shape,
clear, sir. So I'm not getting.
spirillum S shape. So this is the different shape of the bacteria also there. If you given in our book, please read it. This is not a important one also. So anyway, in prokaryote, the primary function of the cell wall is to protect the cell from the internal target pressure caused by the much higher concentration of the protein and other molecule inside the cell compared to its external environment. So that is the main function of the prokaryotic cell wall. And the bacteria cell wall different from that other organism by the presence of chemical composition. You tell me the general chemical structure in plant kingdom. I am asking you plant kingdom. The cell wall made up of which chemical? Most of the plants from algae to angiosperms, the cell wall main chemical component.
Okay, sir. In sheet, like they'll have some sheet cell wall cellulose like
Right,
nucleus.
exactly. Cellulose. Right. Cellulose is the major chemical composition. Right, but in bacteria, instead of cellulose, here the peptidoglycan chemical present. So actually, this is completely made up of the cell wall, which is present immediately outside the cytoplasmic membrane. So this peptidoglycan is made up of polysaccharide. The backbone is consisting alternating in acetyl muramic acid. So in this peptidoglycan, two major chemical present: polysaccharide, muramic acid. N acetyl muramic acid. Nam N acetyl muramic acid and peptidoglycan, sorry, polysaccharide plus N-acetyl muramic acid both together formed as a peptidoglycan. Apart from the acetyl muramic acid, in some of the species, acetoglucosamine NAG residues also present in equal proportion. So this is about peptidoglycan chemical. Actually, in plant, instead of peptidoglycan, plasma cell wall present, cellulose present. But instead of cellulose, here the peptidoglycan present. The peptidoglycan made up of polysaccharide and muramic acid, glucosamine. So this is about cell wall and cell wall components. The peptidoglycan function determine the cell shape. Already told you the cell wall function, it gives the proper shapes to the bacteria. So the exact works carried out by this chemical composition. The chemical composition should be carbohydrate, muramic acid, glucosamine. These are the major components. So the bacterial cell wall, and for the determination cell shape, it is relatively porous and it is not considered to be a permanently barrier for the small substrate. So we can observe here the structure of peptidoglycan for gram positive. Here based on the cell wall chemical nature, the bacteria cell they can be divided two types: gram positive bacteria and gram negative bacteria. Let's see difference between gram positive and negative bacteria. So if you see the gram positive, here the both are have the major chemical peptidoglycan. In peptidoglycan, polysaccharide and muramic acid presents. So here the peptidoglycan is thick, very thick. But if you see the gram negative peptidoglycan, very thin membrane, and the next followed by membrane. Here also membrane also shows shrunken nature. So this is the major difference between gram positive and gram negative bacteria. Actually, this difference of the cell strain base of gram strain reaction is developed by Hans Christian Gram, 1884. Gram positive versus gram negative cells. Gram positive cells, thick peptidoglycan, already told you, and with embedded in teichoic acid. Teichoic acid also present in gram positive bacteria. But in negative bacteria, peptidoglycan layer, outer membrane, lipopolysaccharide, it is very thin layer, and also teichoic acid offset. So this is the cell wall of, are you able to see the diagram? Hello?
No, sir. No, sir.
What happened? Cell wall slide, which slide are you visible to you?
Cell wall is visible, sir.
Now,
Still same page, sir. Yes, sir. Cell wall. Now we can see the diagram.
this tweet. Now which slide? Hello?
Yes, sir.
Cell wall slide.
Nothing, sir.
Now,
Nothing is visible.
Blank, sir.
screen sharing is paused on. Pause my screen sharing. Now visible?
Still it's blank, sir. Yes, we are able to see the peptidoglycan.
Right. Now,
Cell wall.
now,
Differences in cell wall structure.
right. Right. Now cell wall, gram positive, gram negative diagram.
Yes, sir.
Yes, sir.
Right. Already told you, gram positive, gram negative. In gram positive, peptidoglycan thick manner. In gram negative, peptidoglycan thin. You can observe easily in diagram. And also plasma membrane present. Here also plasma membrane present. And lipopolysaccharides and proteins also present. But here lipopolysaccharide proteins not much appearance here. And apart from that, actually the difference first discovered by Hans Christian Gram. Is it visible to you? Difference in
Yes, sir.
cell structure, right?
Yes.
Hans Christian. Are you able to see the Hans Christian name
No, no,
here?
sir.
Are you follow marker?
We are in the still cell wall page, so diagram. Positive, gram positive, or gram negative.
What happened here? Now which slide?
Cell wall, sir. Gram negative and gram positive.
Now,
Differences in cell wall structure, sir.
right. Now are you able to see Hans Christian Gram?
Yes, sir.
1884. Difference in gram positive versus gram negative cells. Gram positive cell, thick peptidoglycan. Just now we observed the diagram. Apart from the thickness, embedded the teichoic acid. In gram negative cell, thin peptidoglycan layer outer membrane of lipopolysaccharide. No teichoic acid. Okay,
Yes, sir.
right. Are you able to see diagram? Cell wall of gram-positive bacteria,
Yes, sir.
right? So if you see the diagram of gram-positive bacteria, as I said, first the layer cytoplasmic membrane or plasma membrane. This is the plasma membrane layer. After that, it followed the cell wall. Already told you the cell wall components polysaccharide, teichoic acid also present. Already told you in gram-positive bacteria, muramic acid also murine also present. This is completely murine. Polysaccharide, these are the polysaccharide. Nowhere there are polysaccharide present. And teichoic acid also present. This is teichoic acid. So teichoic acid present. Is it clear? Hello?
Yes, sir.
So this is bacterial positive bacteria cell structure. If we see the negative diagram, what you observed in negative and positive, you tell me. In positive, teichoic acid present in this diagram. What if you
In positive, it will be purple color,
in
sir.
yes, in negative bacteria there is no teichoic acid. Is there any teichoic acid chain? Cell membrane
No,
common
sir.
in both, murine common in both,
Yes.
and also polysaccharide also common in both. But where is teichoic acid in this? Where is teichoic acid? If you see the earlier side, here the teichoic acid. Are you follow marker?
Yes, sir.
Are you following the marker?
Yes, sir.
Right. Here the teichoic acid, the same structure, same diagram, positive and negative. But in positive, this structure present. But in negative, there is no teichoic acid. That's it. Is
Yes,
it
sir.
clear? And this is also same diagram. The outer membrane. So the first, the cell wall completed. Cell wall mainly, the based on the cell wall components, it is divided into gram positive, gram negative. Actually, the bacteria cell wall not composed with cellulose, it is complete peptidoglycan. The peptidoglycan, main chemical, polysaccharide and muramic acid. These both are main. And in gram-positive bacteria, teichoic acid also present. But in gram-negative, teichoic acid absent. And gram-positive, cell wall is the very thick manner. Peptidoglycan thick, but in negative, thin manner. That is the difference between positive and negative based on the cell structure, cell wall. And next, after the cell wall, outer membrane or cell membrane or plasma membrane. This lipid bilayer is found in gram-negative bacteria and is source of the lipopolysaccharide in the bacteria. The lipopolysaccharide LPS is toxic turn and the immune system, not found in gram-positive bacteria. Outer membrane, this outer membrane, that means which is actually outside the cell wall. Outside the cell wall, this outside the cell wall, tough layer, slime layer, present in only gram-negative bacteria, absent in gram-positive bacteria. So cell wall completed, the outer membrane of the outer layer of the cell wall also completed. Now lipopolysaccharide is not important one. Cell membrane. So this is the order. Cell wall first. Cell wall either side, outer side slime layer or outer layer, inner side cell membrane. Are you able to see the cell membrane slide? Hello?
Yes, sir.
Right. The cell membrane, this is made up of phospholipids and proteins. This is common, not only bacteria. If you take any organism, the cell membrane made up of phospholipids and proteins and carbohydrate. and it forming fluid mosaic pattern. Actually, based on the arrangement, there are proposed several models which in this the most accepted model is fluid mosaic model, which is proposed by Singer and Nicholson, and it surrounds the bacteria and its most important organelle, and it is control the movement of substances in and out of the cell, movement of the any substances inside and outside. It is act as a semi-permeable membrane. Hello or not hello? Permeable membrane. So the structure also not important one. And capsule, slime layer, and S layer. Already told you just now the above the cell wall, the outer layer such as called as capsule or slime layer or S layer also present. The some bacteria have the few material lying outside the cell wall. The cell wall organized, not easily washed off. It's called capsule. The slime layer zone is diffuse, unorganized material that is removed easily. Many of the gram-positive and gram-negative bacteria have regularly structured layer called S-layer structure. The S-layer pattern something like floor tiles and composed of protein and glycoprotein, cell wall, peptoglycan, plasma membrane chemical, protein and lipid, and the slime layer, protein or glycoprotein. In gram negative bacteria, the S layer adheres directly to the outer membrane. It is associated with the peptoglycan surface of gram positive bacteria. It may protect the cell against the ion, pH, acids, stress, high temperature, so on. And so the outer external structure of the cell wall, the most external structure, pili and fimbriae. You can see in diagram, pili air-like structures, fimbriae are air-like structure, which are present many gram-negative bacteria. In gram-negative bacteria have the short, fine air-like appendages that are thinner than flagella, not involved in motility. These are usually called fimbriae. Fimbriae thinner than flagella, not involved in motility. That is called fimbriae. Pilus are similar appendages, about 1 to 10% cell that different from fimbriae in following ways, such as pili often are larger than fimbriae. Pili larger than fimbriae. Fimbriae thinner than flagella, larger than pili. They are genetically determined by sex factor. Conjugate to plasmid are required. F present, that is called F plasmid. F pili absent, that is called F minus. Some bacterial viruses attach the specifically the receptor sex pili and start their reproductive cycle. So the pili, the another type of pili called as sex pili, which is the function of the sex pili involved in reproduction to attach one bacteria to other. And flagella, apart from pili, fimbriae, flagella, the motile bacteria move by use of flagella. Flagella also must have seen in diagram. A thread-like locomotor appendages extending outward from the plasma membrane of the cell wall. They are slender, rigid structure. So this is flagella. So they are all external parts of the cell wall. Pili, fimbriae, flagella. So these are the flagella types. Are you able to see the diagram? Hello?
Yes.
Flagella diagram. Monotrichous. This is monotrichous. Monotrichous. How many flagella appear in diagram from monotrichous?
One.
One. Lophotrochus. How many? And how many flagella and which side? Flagella appearance which side? One side or two sides?
Two sides.
Two sides. And amphibians, but two sides, more flagella. Amphitrachus, of course, like a Lophotrachus, but Lophotrachus more than one flagella present either side. But in Amphitrachus, only one flagella present in both sides. Peritrichus, what you observed in Peritrichus, only bunch of flagella present at one end only, not both sides, only one end. So this is the flagella arrangement. So the flagella is a hair-like structure. Flagellar filaments are made up of subunits of single protein flagellin. If you see the electron trans microscope, you have shown the flagellum composed of three parts. Filament extend from the cell, surface of the tip, basal body, the basal body embedded in the cell, hook. These are the parts of the cell flagella. The basal body of gram-positive bacteria has four rings connected to central rod. A pair of ring embedded in cell membrane. So the flagella, of course, extended from the cell wall, but originate from the cell membrane. Originate from the cell membrane. Flagella originate from the plasma membrane. You can observe in diagram also. This is cell wall. Just below the cell wall, plasma membrane. From the plasma membrane, flagella emergate out. Gram-positive bacteria have only internal membrane system. What is present? Internal cell wall completed, slime layer S-type completed, plasma membrane also completed. What are present inside? Mitochondria, chloroplast present or absent? Already told you there is no cell organelles except the ribosomes. Some other primitive cell organelles I will explain later. And another part of the plasma membrane, mesosomes. The mesosomes are invagination of plasma membrane in the shape of the vesicle tubule lamella. Thus, the cell wall formation, this is maybe involved in cell wall formation during division, play a role in chromosome replication and also distribution daughter cells, and also not given here, and also involved in replication, respiration. So plasma membrane foldings, simple. What are mesosomes? Mesosomes are plasma membrane foldings. Plasma foldings called as mesosomes. Apart from the mesosomes, inclusion body is also present. The variety of inclusion bodies, granules of organic matters such as inorganic matters present in cytoplasmic matter. The inclusion bodies, the function is storing compounds. And synophysin granules, another part of the cytoplasm, synophysin granules, carboxysomes. The synophysin granules are the composed of larger polypeptide containing approximately equal amount of the amino acids. The granules store extra nitrogen for the bacteria. So this is the function of synophysin granules. The synophysin granule also one of the part of the cell. Carboxysomes. Carboxysomes are present in many cyanobacteria. I will explain later about cyanobacteria and nitrifying bacteria and thiobacteria. And what else? Gas vacuole also present. Remember the cell components, inclusion bodies, synophysin granules, carboxysomes, gas vacuole. Gas vacuole, another cell organelle also present inside the bacteria. Is not important. And ribosomes, most important one, ribosomes. What type of ribosome present in prokaryote? 70S ribosome, only 70S ribosome present. These are the smallest and the most numerous cell organelle. Their purpose, protein synthesis for the cell own use. That's why called as protein factory. It consists protein and RNA. They are located either free in the cytoplasm or attached to the RER. So this is the small unit 30 years, large unit 50 years, 50 and 30, 70 years ribosome in eukaryote bacteria. Genetic material side visible or not? Hello?
Visible, sir.
Yes, sir.
Right.
Visible.
Genetic material side visible, just wait. So the prokaryote bacteria cell has no nucleus, but initially someone said of course, there is here actually no nucleus means here remember the word inside bracket nuclear, just wait, nuclear membrane, comma, nucleolus. Right? So are you visible my typing?
Yes.
Just wait. Nuclear membrane and nucleolus. Right? It is genetic material DNA are contained in nucleolide. Here round shaped. Of course, no membrane, no nucleolus, but genetic material present. All that told you earlier. So this is nucleus, bacteria nucleus structure, double-strand DNA, domain protein. This is the microscopic ultra structure of the DNA of bacteria. Bacteria nucleus. Apart from the genetic material, plasmid is addition chromosome, and addition genetic material present. That is the plasmid. These are double-stranded DNA molecules, usually circular. Plasmids are circular, but nuclear in rod shaped. But they are circular in number. These are different type of plasmids there. A different type of parties are F plasmid, R plasmid, coli plasmid, degradative plasmids. The first one, F plasmid, already told you. Some of the sex pili, it involved in reproduction. In that type of bacteria present F plasmid. These are capable of conjugation, transfer genetic material between bacteria to another bacteria. I will explain later conjugation process. In reproduction. Reproduction also very, very important. Okay. Resistance. The resistance are plasmid which contain genes that can build resistance against the antibiotic and poisons. For example, uh, the R type of plasmid can resistance for any type of pathogen, any type of toxins. And coli plasmid which contain the genes that determine the produce the bacteriocin protein that can kill other bacteria. Most of the harmful bacteria have the coli plasmid. The COL col plasmid produce the toxin. R plasmid resistance to toxin. Col plasmid produce the toxin and cause the disease and even kills the organism. Degradative plasmid which enable the digestion and unusual substances. So this is the about the bacteria cell structure. Any doubt in bacteria cell structure? Hello?
Nothing, sir.
But you should read in order. First, bacteria components. You should list out what are the bacteria components, and one by one you have to explain. First of all, cell wall. In cell wall, gram positive discuss, gram negative discuss, and cell wall chemical composition discussed. And next, above side, outer side, capsule, S-type wall, they are present. And inner side, cell membrane. After that, some of the cell organelles. But most important one here, the nucleus. Nucleus such as means the genetic material, extra genetic material plasmid. That's it. Is it clear?
Yes.
Hello? Right. Now the bacteria reproduction. Reproduction also very, very important. What is reproduction first? What is reproduction? Hello?
Reproduction.
So passing of genetic material and division of bacteria, production of bacteria.
Right, that's it. The simple genetic material transfer or fusion of genetic material. That is that is called as reproduction. Actually, how the genetic material exchange, how the genetic
Binary
material
fission.
fuse. Binary fission, not exchange genetic material. There is no exchange in binary fission. In binary fission, just share the genetic material, divide the genetic material and form. And that means exchange means fusion. Fusion of actually in reproduction three types: vegetative reproduction, asexual reproduction, sexual reproduction. I am asking you about only sexual reproduction. What is sexual reproduction?
Sexual reproduction, where gametes.
Right, gamete formation, fusion of gametes, right?
Yes, sir.
But in bacteria, no gamete formation. Remember, no gamete formation,
Okay.
no fusion of gamete. Although this considered as a sexual reproduction, why it is considered? Even not formed any gamete, any fusion. Just earlier you said material exchange. What happened during the sexual reproduction in higher organisms? Gamete formed, the gamete fused. While the both gamete fused, the both gametes genetic material exchange each other. Right? Hello?
Yes, sir.
Hello? But in bacteria, only genetic material exchange. Only genetic material exchange without formation of gamete, without fusion, remember. So this is done by three methods. One is conjugation method, second one is transformation method, and third one transduction method. Transduction method. This is sexual reproduction. But another one's vegetative reproduction, binary fission. What is binary fission? In name itself given binary. Binary fission. Hello?
Binary.
Binary fission. Simple. Hello? Bi means
Two.
So by means two, sir. Like where cell
two.
must copy its genetic material.
Yes. So one bacteria cell forms into two bacteria cell. So that is called binary fission. If you see diagram, just wait. It is. Are you able to see diagram? Just wait. Somewhat difficult. Are you able to see? Hello?
No, sir, nothing.
Share. Where is bacteria? So, okay, we will complete to next class. Only remain reproduction, economic importance. Already told you. You can read yourself. Okay.
Yes, sir.
Binary fission, sexual reproduction, and virus. These are all complete in my next class.
So what
Meanwhile,
about fungi?
fungi also. Fungi also complete. First, next class, virus complete and bacteria reproduction complete. That is third class. In fourth class, I will explain fungi general character, like a alga general character.
Okay, sir.
Meantime, you have to read about the algae type studies. Cara,
Yes,
Falvax,
sir.
Pseudogonium. Individually, you can read.
Yeah, sir.
And after that, while you're reading, if you have any doubt, we will discuss and ask me, and I will explain in my next class.
Yes, sir.
Okay?
There are a lot of types of algae, sir. Blue algae, red algae, like.
Yes. These are comes under the concept of algae general character. Okay?
Yes.
Right. Thank you. Thank you all.
Okay, sir. Have a nice day, sir.